All right, today we're going to talk about the ubiquitin proteasome pathway and how this pathway can lead to protein degradation. Now, so far we've talked a bit about how proteins can be modified, how we can covalently couple carbohydrates onto them, what that can do, how we can add on lipids onto a protein, and how that can target it to the membrane. Uh, we can covalently modify proteins by changing disulfide bonds. We can phosphorylate it or add small molecules. So we've talked about a lot of different ways that we can modify a protein after it's already been made. And ubiquitin is just simply one of those modifications that we can make. So how this all works out is we have something called an E3 protein. E3 is a class of proteins, not a specific protein. So within this class of E3s, we have individual specific E3 proteins. Each one of those specific proteins will target a particular target protein. Okay, or it will search for and bind to a particular target protein. And it typically does this by recognizing a short amino acid sequence that is present within that target protein. And I like to refer to those short sequences as tags for proteins. We will see tags come back in other contexts as well, such as targeting a protein to go to a particular compartment, or to be trafficked to a particular area, or to have various different things happen to it. It is all done through recognizing a short amino acid sequence. So we have these E3 proteins. They are searching out target proteins that have the recognition sequence on them. We then have an E2 protein that recognizes E3. And again, E2 is a class of proteins. There are many of them, and each specific E2 protein will target a specific E3 protein, okay? So this allows us to put in a level of control by controlling how much of that specific E3 we make and how much of that specific E2. We can regulate how much of this target protein is being ubiquitinated and how much ubiquitin is being added on. So E2 has ubiquitin represented as this green ball. E2 will then transfer it onto the target protein, and we'll talk about how it does that here in a moment. And once it transfers one on, it can transfer another onto that ubiquitin, and yet another, forming a chain of ubiquitins, right? And the longer that chain is, the more likely it is to be recognized by the next player in line. In this case, we're going to be talking about the proteasome. All right, now it'd be nice if it was just this simple, but we are going to add one more layer on. What we have here is something called a class of protein called the E1 protein. Its job is to take ubiquitin, use the power of ATP in order to couple that ubiquitin onto itself. Some of that energy from ATP is retained in here, and it can be used to facilitate the transfer on the E2, and that energy is retained even a bit further, allowing it to be transferred onto its target protein. Now, you might ask yourself, why don't we just load ubiquitin directly onto E2? Well, as I said, this is about control. So if we can control how much E3 protein there is and control how much E2 protein, if we can also load that ubiquitin on with E1, we can now control how much E1 protein we make in order to fine-tune how much E2 is actually charged with ubiquitin, which is another level of control. So this is all about being able to fine-tune so we can precisely say this particular type of target protein, we want to get rid of all of it, or we just want to get rid of a little bit of it, right? So this gives us a lot of control, kind of like a dimmer switch. Now, ubiquitin here we have represented as a small green ball, much smaller than the actual protein itself. And while that's true, recognize that ubiquitin is actually its own fully formed protein. Has 76 amino acids long. You can see that it has a beta sheet in here, a couple of alpha helices, some loops present, and this big long carboxy terminal tail. So it is a large structure that we are adding on. So this is not just a simple little phosphate group that we're adding on. This is a big modification. Now, how we add it on is on the carboxyl terminal end, we have carboxylic acid. If you remember how proteins are put together, we can add a carboxyl terminus to an amino group and form a peptide bond. And that is what we do, except for what we do is we add it onto the amino acid or the amino group present on lysine residues on that target protein. So that target protein probably has one or several lysine residues that have an amino group sticking out. Since it is hydrophilic, it tends to point outwards. We essentially take the back end of ubiquitin and form a peptide bond. And this is a peptide bond just like anywhere you would see in the backbone, except for it just happens to be present on a lysine. So to distinguish that is slightly different, we're going to call it an isopeptide bond. But the peptide bond itself behaves in the unique ways that peptide bonds do behave. So we are adding ubiquitin onto lysine residues on the target protein. Well, as you may have noticed up here, ubiquitin has several lysine residues of itself. And this is what facilitates the formation of these chains of ubiquitin, right? So we can add a ubiquitin on here and or a ubiquitin onto there or one onto here. So we can form these long chains, and notice that there is some diversity about having several of these lysine residues. That's going to come back and become important. Now, typically what happens at this point after we have made this chain of ubiquitins on this target protein is that target protein is then transported to something called the proteasome. A proteasome has proteins that specifically recognizes chains of ubiquitin. That will take that protein, it will load it into the inside of this barrel-like structure. I just want to point out here that it is very easy for students to confuse the proteasome with the chaperone, which is another barrel-like structure that refolds proteins. This is a barrel-like structure that destroys proteins. So just be aware that there's two different things out there, and they look very, very similar in structure. But on the inside of this, on the inside of the proteasome, we have several different proteases. And those proteases fall into the same classes that we've seen in our mechanisms of how trypsin works or how chymotrypsin works, that those classes of serine proteases. It has several of those within the inside of this, each one targeting a specific different amino acid, right? So some things are targeting hydrophobic amino acids, some might be looking for basic amino acids, and then it'll introduce a cut in the backbone just next to that. What this will do is create several short peptides, and it will free up the ubiquitin that can then be used for other things. This is a process that does require ATP along the way. Once we have these peptides, we can further break those peptides down with proteases that are just present in the cytoplasm. Those proteases can't break down this big protein because it's too tightly folded. So unraveling it is part of what the proteasome does. Now, if you recall, there can be different ways that we can, or there are different lysine residues present on ubiquitin. This leads to this paradigm that how we form these chains can make a difference of what happens to that protein. So here we're going to focus in on this proteasome degradation pathway, where we have a target protein, lysine residue, we get a ubiquitin put on. After that, all the subsequent ubiquitins that are added on are all added on to lysine 48. And it'll be added on to lysine 48 over here and lysine 48 of the other. So it's a link or a chain of lysine 48 linked ubiquitins. That's going to target it specifically to the proteasome, to where these other pathways have separate functions. So although targeting proteins for destruction is the main use of ubiquitin, there are some other things that are happening here as well. Here we have T lymphocyte control. T lymphocytes are assassin cells in our immune system. They target cancer cells or virally infected cells and directly kill them. That is something that we need tight control over. So we have ubiquitin pathways controlling parts of that mechanism. Up here we have a separate system that is involved in immunity signaling. Again, something specific for our immune system, which requires specialized signaling. And here we see chains of lysine 63 residues being modified. Finally, here's a last one where we have this process being involved in cell division, where we have target proteins that are being ubiquitinated in chains of lysine 11. So these different patterns are what's being recognized by different players downstream. The proteasome is looking specifically for this chain of lysine 48 linked. All right, so to give you an idea of how the system might work and what an outcome of this might be, we're going to talk about cells going through cell division, right? So we have that long interphase where we replicate the DNA and then we proofread it. And then we go through that process of mitosis, which has different phases such as prophase, metaphase, anaphase, telophase. Well, the thing that controls a cell going from interphase into prophase is the activity of this thing called CDK1. This is a protein kinase. It phosphorylates other proteins, specific other proteins, and that phosphorylation event is what allows it to progress into prophase. Now, CDK1 only works when it is bound to cyclin B. Okay, so the way we can control this process is in interphase, we don't make cyclin B. And when we are getting towards the end and we know we're ready, we start making cyclin B. Thus, we start turning on CDK1, and that will allow us to start progressing into prophase towards metaphase. At the same time, we are also starting to ubiquitinate cyclin B, right? And if we add on just one or two, that's no big deal. But as we start getting longer and longer chains, that is going to target cyclin B to the proteasome, and it is going to be broken down and destroyed. If we separate cyclin B from CDK1, CDK1 now becomes inactive. And you can see that it becoming fully inactive is here at the threshold of metaphase. Remember that metaphase plate where all the chromosomes are lined up in a row. Once we move past there, then we can go into anaphase, telophase, go through another round of interphase where we then start producing cyclin B again. We'll make it, ubiquitinate it, then destroy it. You can see that this is kind of acting as a timed fuse, so to speak. We put it on, we put the fuse on, and then we light the fuse. Now, to give you a little further insight into how the proteasome actually works, we have these large ring structures made up of individual alpha or beta type subunits forming these ring structures with a large empty interior. Within the beta rings, we have the proteases. We have 3 different ones per ring. So the inside the interior of this, we have 6 total different proteases working. And we have these large cap-like structures on the top and bottom. Again, these caps are very similar to the caps that are present on chaperonins. So again, it can be something that is very easy to confuse the two, to try and keep them separated. One destroys, one refolds, renews. So we have our target protein, we have a chain of ubiquitins. In this cap is the proteins that recognize the ubiquitin chain. Once we recognize that chain, we can free the ubiquitin from it, and we have the proteins that act as a machinery to start unwinding the protein, feeding it single file into the interior, where it can then be cleaved by the proteases into these short peptides. These short peptides actually have activity of their own. You can just break them down and recycle them, but our immune system actually utilizes these short peptides to help us recognize what our own proteins are, and we can talk about that much later. So here I've given you an introduction into the ubiquitin pathway, focusing in on this ubiquitin proteasome pathway.